And thank you to all of you joining here today for the presentation. I'm Dave Butler. I'll be talking about chemoenzymatic ligation, advancing next-generation manufacturing technologies for siRNA and sgRNA. These are my disclosures. And for a presentation overview, I'll give an introduction about chemoenzymatic ligation, and then we'll dig into some work that's actually been going on at Hongene around siRNA synthesis. There'll be a case study, which is divalent siRNA, or containing XNA, and then controlling the cost of manufacturing with a case study, which is crude to purified, or C to P siRNA. And then we'll touch on some work that we've done with our engineered thermostable T4 RNA ligase. And we have an exciting update also on the translation of the technology into our manufacturing suites to support clinical development for an siRNA. And we'll finish with some CMC regulatory considerations and finally conclusions. Okay, introduction. So there's really the impetus for this is largely the large market potential for siRNA drugs. And any one of these 6 drugs in the table has the potential to reach about 10 million or more patients in the US alone. So all of these target cardiometabolic genes associated with cardiometabolic disease. And a few of these could be approved by 2030, and our research suggests that more than 10 tons of API with sufficient market penetrance of these could be required by the early 2030s. And that's going to put a strain on existing methods of manufacture. And so that's the reason that we're here today talking about this. And then on the right-hand side, we have kind of a fun graph showing the potential life cycle trajectory for siRNA therapeutics, assuming that it mirrors what we've seen for mAbs. And certainly clinical pipelines suggest that this could be the case, which is really very exciting. And this is shown here for siRNA, but you could equally draw a similar graph for other oligonucleotide modalities, I think. So at Hongene, we think about oligosynthesis technologies in terms of 3 generations. And firstly, on the left-hand side is generation 1. That's the fully chemical method using phosphoramidides. And we're standing on the shoulders of giants here. It's the current paradigm for manufacturing, and it served us extremely well until this point. However, there are some drawbacks. Relatively speaking, lower purity, lower yield, and not very good sustainability. It generates a lot of waste to make oligonucleotides using this method. Generation 3 technologies, meanwhile, are those that are fully enzymatic, and these rely on processes that you see inside cells, so like DNA replication, mRNA transcription. And it's still quite early in the development of these processes, although companies such as Codexis, N+1, and others are making some really good advances in this area. And there's no reason to expect that with sufficient engineering of enzymes and processes that this won't be where we all end up for oligo manufacturing at some point in the future. In the middle of these is generation 2. So this is chemoenzymatic ligation of oligonucleotide fragments. And because this relies on generation 1 technology to synthesize the oligonucleotide fragments, it means that it's ready for prime time today. And in our hands, the advantages are clear. Always higher purity, higher yield, better, more cost-effective, more scalable, and better sustainability. So we think that this process is going to play a huge part in oligonucleotide manufacturing for at least the next 10 years and beyond. Hongene's chemoenzymatic ligation the platform consists of 2 processes. First of all, on the left is sticky end ligation, which is perfectly suited for RNA duplexes like siRNA. And this was first described by Khurana more than 50 years ago. And in our take on this process, we have 4 oligonucleotide fragments that are hybridized, and that results in 2 duplexes that have complementary overhangs that hybridize in this so-called sticky end cohesion step, and then the junctions between the oligonucleotide fragments are ligated with an enzyme to generate the siRNA duplex directly, and that is purified by non-denaturing column chromatography. So this obviates the requirement for rather tedious and expensive purification of single strands separately, as is the case for a generation 1 process to make siRNA. And then on the right-hand side, we have splinted or template ligation, which is perfectly suited for long RNA molecules such as single guide RNA and pegRNA. So in our take on this process, we have a certain number of oligonucleotide fragments and a certain number of complementary DNA templates, and it depends on the length of the molecule. And these are hybridized, and then the junctions between the oligonucleotide fragments are ligated, and the DNA templates are digested with DNase 1. The crude single guide RNA is purified by denaturing column chromatography to generate the single guide RNA to the pure material, and it's really highly pure using this process. So the mechanism on the left-hand side is shown here. So we have, in the first step, reaction of lysine on the enzyme with ATP to generate this phosphoramidate intermediate that then undergoes attack by the 5' phosphate to generate a bridged diphosphate, which then undergoes attack by the 3' hydroxyl group on the adjacent fragment to generate the ligated oligo product. So it's a 3-step process. And there is some important features of ligation. So firstly, some shortmers are rejected. They're just not, either they're not good substrates for the ligation reaction or they're not substrates at all. The process can be optimized very easily through changing the buffer, the temperature, reaction time, and engineering the enzymes, etc. And when you have a prochiral 5' thiophosphate as the substrate, you generate an RP phosphorothioate diester in the product. And that's an important consideration if you're making antisense oligonucleotides that contain phosphodiester's using this technology. Okay, so we're going to jump into some actual work that we've done. And first of all, this divalent XNA containing siRNA. And this technology was invented at UMass, and the monomers here, you can see on the bottom right, the XNA monomers contain an additional methylene group at the 5' position that confers stability against nucleases, and that improves the durability of these molecules and the pharmacology. And this paper on the bottom here, the reference, Yamada et al. Was awarded the OTS Paper of the Year a couple of weeks ago. So scientists at Broad, UMass, and Harvard recently identified a divalent siRNA development candidate for prion disease. And Hongene was honored to be selected as the CDMO for manufacturing this material. And we did it with solid phase synthesis, so generation 1 technology. But we were obviously very curious as to whether we could apply chemoenzymatic ligation to synthesis of this very interesting construct. And so we selected a model divalent siRNA, which is shown below. The sequence is hidden, but the chemistry information is all there. And so we consider this to be like a stress test of the technology, right? It's a kind of interesting and difficult molecule, as you can see. And this is the chemoenzymatic process design. And here we're applying what we call our P2P process, and that means we're purifying the oligo fragments and we're purifying the ligated siRNA. So you'll notice that there's a lyophilization step for the oligonucleotide fragments. We typically do this for small-scale reactions where we're looking at case studies, but the lyophilization step can be removed when you go to large scale for manufacturing. And the overall reaction scheme is shown here on the right-hand side. The oligonucleotide fragment purity is measured by IPRP HPLC-MS, and following synthesis, you'll see the purity of these fragments is really very high. It's above 93% across the board. The yield is kind of moderate. It's about 40% on average, but that's what you might expect from this reaction, which is the P2P process, or purifying the fragments. The HPLC method here, it's a reasonably good method, right? We're able to have good resolution of closely related impurities like N-1, N+1, PO, PS, etc. And we're able to resolve those and quantify them. And so following synthesis of these fragments, we mix them in precise stoichiometry and then feed them into the ligation reaction. And then this is what you see on the other side. So this is a denaturing IPRP HPLC-MS method. So we're looking at the two strands together, right? And they're resolving on the column. So the first thing that you can notice by eyeballing the crude here, which is the blue HPLC, is that there's a very high fragment conversion. And in fact, the reaction is quantitative in the limiting strand. Then we also have very pure siRNA product. It's approaching 97%, and this is better than we would see for a similar molecule made with solid phase synthesis, where we'd be looking more at the 90% purity level. And the yield is moderate. Again, that's expected. It's the P2P process. Digging down a bit on the impurities. Impurities A through E here are all fragment-related impurities. So these are, you see these in the crude product, but not in the purified material. So these are purged easily. Impurities F through, I think that's J. F through J, meanwhile, these are found in the crude and they persist into the purified material. However, these are all solid phase related impurities, which is important, and there are no new ligation related impurities associated with the ligation process, which is a question we often get. Lastly, all of the impurities are below the 1% ID threshold that's suggested by Capaldi et al. In the seminal OSWG paper referenced there. Now we look at the non-denaturing SEC. The purity is mirroring what we see for the denaturing column chromatography. However, we have this rather irritating little shoulder on the right-hand side, and we hypothesize that that could be due to incompletely duplexed product. So we call it a half duplex. And so we did a simple study here, taking excess of the sense strand and mixing it with a limiting amount of the antisense strand to generate the half duplex deliberately. And indeed, this overlaps with the shoulder, lending some credence to the hypothesis that it could be the half duplex. Hongene is now licensed to sell XNA monomers and oligonucleotides for research purposes. So if that's of interest to you or your colleagues, please download the flyer here, come and talk to us at the booth. We can hook you up with some of these very interesting monomers and oligos. Okay, so for manufacturing these large volumes of cardiometabolic disease drugs, the cost of manufacturing is going to be paramount for getting good market traction and getting people to, getting more people to take these medicines. So we think there are three key levers for achieving this. Firstly, removal of chromatography, optimization of the process. Secondly, engineering enzymes. And thirdly, improved fragment manufacturing technologies. So I'll not talk about the fragment manufacturing technologies today. Suffice to say that the way we manufacture these now is in these flow synthesizers, which are scale limited to about 10 kg. And so we're actively investigating better methods that are more scalable and cost effective. Okay, so removal of chromatography. And so we've described the P2P process already. The C2P process is where we remove column chromatography from the oligonucleotide fragments. So that's crude to purified. And then crude to crude is what we call C2C. That's under active investigation. We think that our engineered enzyme is going to play a huge role in this in the future. And then you can think that even further down the road, we can get rid of lyophilization as well and move to a liquid drug substance. And this would be a much more cost-effective method for large volume manufacturing. Okay, so the C2P case study. And again, we have a lyophilization step for the oligonucleotide fragments. It's on small scale. This we remove when we go to large scale. And the reaction scheme is shown there on the right-hand side. And the target molecule here is a clinically relevant siRNA. And it's a model sequence. But the sequence is hidden and the specific chemistry is hidden. But it contains all of the normal modifications that you find in these molecules, like 2'-O-methyl, 2'-fluoro, phosphorothioates on the ends, phosphodiester backbone, and a GalNAc on the 3' end of the sense strand. And the fragment purity is measured again by IPRP HPLC-MS. And actually, the purity of these oligonucleotide fragments, even in their crude form, is really good. It's above 88% across the board. And it surprised us a bit. But these molecules, they're just shorter than the full-length sequences, and you see much better purities in general for the shorter sequences. And the yield here is about 68% on average for the 4 fragments, and that compares to about 40% on average for the P2P process. Again, these are mixed in precise stoichiometry and fed into the ligation reaction. This is what we see in the denaturing IPRP HPLC MS method. So again, very high fragment conversion just by eyeballing the crude. Again, very high purity of the siRNA duplex, higher than you would see for this molecule manufactured using generation 1 technology. And the yield is much better. Typically, we see 40 to 50% yield for the C2P process. Digging down a bit on the impurities again. A and B are fragment-related impurities. These don't persist into the siRNA product following purification. The other impurities, meanwhile, in black, C through H, these are found in the crude and in the purified material. So these persist. However, they're all solid phase related. There are no new ligation-related impurities. We do see this M-20 Dalton impurity, which is often accompanied by an M-2 Dalton impurity. And we see these following lyophilization and on storage in solution, and these are associated with degradation of 2'-fluoropyrimidines. Lastly, importantly, all impurities are below the 1% ID threshold suggested by Capaldi et al. And this is typically what we see, in fact, is that we don't see impurities above 1%. Looking at the non-denaturing reverse phase method, the crude product, you can clearly see single strands, residual fragments, and then these are totally removed in the purified duplex, the purity of which mirrors what we see for the denaturing column chromatography. Oligofragments, these are an important focus point for us for impurity control. And there are 2 reasons, really, for improved purity for generation 2 versus generation 1. Firstly, some oligo fragments just aren't good substrates for ligase. Right? They don't have the right hybridization characteristics, or they're just not, they can't be substrates at all, as is the case for these impurities A through E in the table. This is the HPLC from the case study, fragment 1. So these impurities all lack a 5' phosphate, they're shortmers, and so they can't possibly ligate, so they don't persist into the siRNA following purification. Secondly, the impurities that do persist, such as F through L in the table, these persist into the crude, sometimes into the purified material. These are all solid phase related impurities. And as I mentioned earlier, there's just a lot less of these impurities compared to what you would see for a full-length sequence synthesized using generation 1 technology. And we think that this is simply because there's less opportunity for these impurities to form and accumulate in these shorter sequences. So the fragments here and the convergent synthesis really is the feature of this technology in terms of the purity and the efficiency of the reaction. This is a table to summarize exemplary siRNAs and single guide RNAs synthesized by ligation. And so the siRNAs on the top, these were synthesized by P2P. So you get a 97% purity there, but moderate yield, typically 20 to 25% yield. And the C2P process, we typically see 40 to 50% yield for that. These two lines on the bottom are longer RNA molecules. So these is a single guide RNA. This is a 100 mer, and a PEG RNA, which is a 161 mer. These are both from the literature. These are synthesized by splinted ligation. The purity of these is exceptional. I don't have time to go through a case study of this today, but if you are interested, we did go through this at the US TIDES conference, and you can email me, I'll send you more information about that. Just going to touch on the engineered thermostable T4 RNA ligase. So the rationale for engineering the ligase for thermal stability is that you can facilitate ligation at higher temperatures, and thereby you can reduce secondary structures that would otherwise cause the enzyme to engage non-optimally with the system. And you can enhance the ability of the ligase to deselect for oligonucleotide fragment impurities, and thereby get mitigation of incorrectly ligated byproducts. And we believe that this method, this process with engineered enzymes is going to be key to enabling the C2C strategy further down the road. And my colleague, Chris Lee, presented on this in detail at the OTS conference a couple of weeks ago. And again, you can email me or email Chris. We'll send you more information on that. But in the interest of time, there's not enough time to go through that today in detail. Suffice to say that we have data, we've generated data to support the rationale, I think. Okay, very exciting update that we're now using this technology in our manufacturing suites to support clinical development. And this example here is DNV001. There's a link to the press release down there on the bottom right. So this is an siRNA that targets PCSK9, and it contains all of the modifications that you would see in these cardiometabolic disease siRNAs that were in that table, so of methyls, fluoros, GalNAc, etc. And we manufactured the non-GMP batch for our partner using the C2P strategy, but we manufactured the GMP batch using the P2P strategy. And the purity is actually comparable. In both cases, it's better than generation 1 technology. So 95% purity or above. Finally, some CMC reg considerations. We manufacture the oligonucleotide fragments under GMP today, but there is a strong case, I think, to be made to justify these raw materials as regulatory starting materials, and there's we're all discussing this about what the plan of attack should be. Secondly, control phosphorothioate stereochemistry. So you want to be able to demonstrate that you have comparable stereochemistry going from generation 1 to generation 2 processes if you're trying to justify a switch, or you need to know at least what the distribution is. And then you also should be measuring this to demonstrate that you have batch-to-batch reproducibility. So HPLC and NMR are both capable of differentiating between diastereomeric distributions. And so we're investigating both of these methods, and we have a collaboration that's ongoing with Bruker for the NMR component of this. In fact, Victor Beaumont is talking in the other spotlight presentation on this topic right now. ID, purity, and impurity control. So, as I mentioned, we're using a denaturing HPLC method for resolving the antisense strand and the sense strand and all of the impurities, because we don't have the ability to do it on the single strand separately, right? We're not making the single strand separately. So that's different. So for generation 1, the purity, the quality control of the single strands is paramount, right? That's the focus for control. But for us, for generation 2, the focus for control is the oligonucleotide fragments. And then the control of small molecule ligation reagents. So this is something new, and you need to demonstrate that you know what's going on there. Finally, there's a new CQA, which is the residual enzyme. And you need to have sensitive, specific methods for measuring and quantifying this. So we have those. We use ELISA and mass spec for that purpose. So these are just some of the regulatory considerations that you will be talking with your CDMO partner about and with regulators. However, it's just paying lip service to it, really. Any one of these topics could be an entirely standalone presentation in its own right. Finally, some conclusions. So this work is driven by the market demand for siRNA drugs, and that market is anticipated to grow significantly over the next decade. We believe that this demand will be met by adoption of chemoenzymatic ligation technology. The advantages are clear: always better purity, yield, scalability, it's more cost-effective, and it's just a better process for manufacturing these things at scale. It's a versatile technology. It supports siRNA, single guide RNA, and other oligonucleotide products. And the priority for us right now for enabling large-scale manufacturing is removal of chromatography, engineering enzymes, and in better processes for oligofragments. Very exciting. We're in our manufacturing suites with this today, and we're getting our customers switched from old process to new process, and some customers are coming in and wanting to start with the generation 2 process right off the bat. And we see no significant regulatory hurdles prohibiting wider adoption of the technology. So I just get to talk about this, but these are the people that do the actual work. The Hongene oligo team, led by Jim Yang, and the Hongene biocatalysis team, led by Minjie Wei. So thank you. I want to thank you all for your kind attention, and visit us at the booth. There's a couple of QR codes here, and if there's time for questions, I'd be happy to take a couple of questions.
Thank you for that. Any questions?
Thank you very much for inspiring speech. I was wondering, even so you mentioned that there is a simplification downstream process, but the presence of T4 enzyme, plasmid DNA, and supposedly genomic DNA contamination, how it affects like the downstream and regulation from GMP point of view?
So you're asking about possible contamination in the
with
enzyme
protein,
itself?
yeah, and with part of bacteria after lysis.
Yeah.
Plasmid or
Yeah, fortunately, we have quite a lot of experience in the mRNA synthesis area as well through IVT. So we do have very sensitive methods for measuring these characteristics on for the purified enzyme. And so, yeah, those are well controlled.
So I would suppose that you implement downstream for mRNA for siRNA in this case to get rid of protein.
I'm sorry, I didn't quite catch the question.
Because you're supposed to get rid of protein and genomic DNA or plasmid or whatever, so you need to implement the mRNA downstream here for siRNA if you ligate them instead of like chemical synthesis. Am I right?
Not quite sure I understand. So
the downstream purification might be implemented the same way like for mRNA to get rid of protein and
oh yeah, sorry,
yeah,
of course, yeah. So
it's a bit of balance between chromatography and I mean it's not just simplification in this case.
Yeah, well, we do have a quick process for removing the residual enzyme. Yeah.
Okay, we'll just take one more question over here.
Hi, great presentation, thank you. Are you looking for a suite of ligases, or can you do all of this with just that one enzyme, the thermostable enzyme?
So we do have a suite of ligases that we've engineered for thermal stability. We're also engineering for other characteristics. But in general, you can get these reactions to work with even the most basic enzymes. So with the wild-type enzyme, it will work. However, it's not optimal. You can always make better enzymes, right, to optimize the reaction. Yeah.
I think we had another question, or do one more.
Thank you very much. Love your presentation. Just a question about the costs. So I'm fully sold on the technology. I believe this is the way forward. But now that you're going into scale up, do you start seeing benefits also on the cost? And can you maybe ballpark how much is the benefit on the cost compared to the full SPOS?
Yeah, so the COGS, certainly we see an improvement even at small scale. So even at 1 kilo scale, we see an improvement there. The length of the process is about the same in terms of the time. But then the real advantages are seen when you go to much larger scale. So we have modeled it out, and we're seeing like double-digit percentage improvements as you go to much larger scales. But of course, we haven't done the ligation at like 100 kg scale yet. But our models suggest that the cost benefits will be significant larger scales.
Thank you.
Nice work, David. I wanted to know if when you were talking about the efficiency of the ligases, do you make a screening process as to where you actually make the ligation when you're setting up a synthesis? Like walking the site of ligation along the sequence to see where you get the best efficiency? Yeah,
it's an empirical process. We do have an algorithm too that will predict what the best site of ligation could be. But of course you need to generate some molecules and try a bunch of stuff out. There's not so many permutations to try out, so yeah, it's largely empirical at this stage.
It would be great if you put that algorithm in the public domain. It wasn't a question, it was just a comment.
Thank you. Well, unfortunately, we are out of time, but I'm sure David will be around to catch throughout the conference.
Absolutely.
Or, of course, bypass the Hongene booth at 427. Could I get another round of applause for our speaker, please?
Thank you.